In this video, we'll be talking about the actinomycetes and viruses. If you have watched the previous videos, you must be knowing that we are covering various topics of soil microbiology. And if you have not watched the previous videos, their links are provided in the I button and in the description box. So in this video, we'll be talking about some important facts related to the actinomycetes and viruses that can be asked directly in various competitive exams like JRF, SRF and ASRB net examinations. So we'll be focusing on important facts and also on life cycle of viruses, that are Litic Cycle and Lysogenic Cycle. Okay, so hello everyone, welcome to the channel Agricultural Studies. My name is Divagar Khatri, and without any further delay, let's get started. So let's start with the actinomycetes. The very first thing that you should know is taxonomically actinomycetes are like bacteria which possess aerial hyphae like fungi. It means the actinomycetes have some properties like fungi and also some properties like bacteria. So it is a link between the bacteria and the fungi. So taxonomically, we are saying actinomycetes are like bacteria. Why? Because these organisms share characteristics of both bacteria and fungi. In bacteria, we can say their cell size, their structure and mode of multiplication are like bacteria, and as of fungi, they form various branches, or we can say they form hyphae like fungi. So actinomycetes are somewhat like fungi and also like bacteria. They are also called the ray fungi. You should know this term. Ray fungi is related to the actinomycetes. Second or third point is they grow as filamentous mycelia and they form spores and they form bacterial size hyphae. Their hyphae are not so long, their size is 0.5 to 1 micrometer in diameter. It is micrometer, it's mu meter, that is micrometer in diameter. They are in an evolutionary phase between the two. Between two, what are these two? Bacteria and fungi, right? Next thing that is very important for various competitive exam is they are next to bacteria in number and are widely distributed in soil. After bacteria, which one is the most abundant in the soil? We'll answer actinomycetes. The first one is bacteria. In bacteria, the bacillus are most abundant. Then second one is actinomycetes. Then third will be fungi. And fourth will be algae. This sequence can be asked in various examinations. So please note that sequence in your notebook. And actinomycetes are most abundant in dry soil. Remember this, they are abundant in dry soil, then in wet soil, and more in the grassland and pasture soil than in the cultivated soil. So they are more abundant in the undisturbed soil, or undisturbed soil like grassland and pasture soil. And they are more abundant in tropical soil than in soils of temperate regions. In tropical region, you know that the temperature and moisture availability are good, and more in the surface than in the subsurface. So in the topic related to the actinomycetes distribution in the soil, I have collected the various facts from various books. So these facts are directly asked in the various examinations. So please kindly learn these facts that are really important. Whenever I highlight any line, that means in my perspective it is very most important. So like bacteria, they are more common in the neutral to slightly alkaline soils, and they are aerobic organism. That means they need oxygen for their life activities, and therefore their number is less in the lowlands, okay, or we can say they are higher in the uplands, okay. So please remember these terms, the actinomycetes, where actinomycetes can be found more and where they can be less, okay. As they can withstand drought conditions very well, they occur more frequently in the soil underlying dry spells. Soils with dry spell. Do you know what is meaning of dry spell? If you have learned something called the dryland agriculture, there are definitions of dry spells. It is a continuous days. These are continuous days, or we can say dry spell is a continuous days in which the dry spell is a continuous days in which the rainfall has not occurred, right? Continuous 4 to 5 days, or we can say less than 1 week. The time period in which the rainfall has not occurred, which should be less than 1 week, it would be called dry spells, right? So in dry spell, we can say the actinomycetes are more. Okay, learn these topic, learn these facts. Please learn these facts. They are really important. So what are the species of actinomycetes that are abundant in the soil? As I said, in bacteria, the bacillus are most abundant. So in actinomycetes, the streptomyces are most abundant. Please remember this species of actinomycetes. And other common species are Micromonospora, Nocardia, and Thermoactinomycetes. But because I have highlighted this, I have not mentioned here, but I have highlighted this because the actinomycetes are most abundant in among the all actinomycetes. Please remember that. Okay. Next is actinomycetes are nutritionally heterotrophic. That means they are dependent on other food supply and highly adaptive to the degrade a wide range of organic substances, particularly those which are difficult to decompose by other organisms. What are the other soil organisms? The bacteria and the fungi. The things that are not decomposed by the bacteria and fungi, then they are discomposed by actinomycetes. Please remember that. Any compound that are being decomposed by the bacteria and fungi, but some part of that is not being decomposed, like it have some complex substances that are, so the bacteria or fungi are unable to decompose that, so the residual compound will be decomposed by actinomycetes. Please, so the actinomycetes are more stronger for decomposing things, right? So remember this topic. They are not photosynthetic. That's why we have said that they are nutritionally heterotrophic. Okay. Next is the activity of these organisms, particularly that of the streptomyces, is below the pH 5. The activities of these organisms, streptomyces, particularly talking about the streptomyces, they are more active at in acidic pH. Okay, optimum temperature are of mesophilic range like 28 to 37. These ranges are important for the examination. Please remember that thermophils are more common in the manure and compost. In compost heap, which actinomycetes are more common, we will say the thermoactinomycetes. Okay, please note these points, they are important for the examination. Next, these organisms start functioning when easily decomposable fractions like the sugar, starch, etc. are used by the bacteria and fungi. I was talking about this point before, right? When easily decomposable things are completely decomposed by the bacteria and fungi, then the actinomycetes come. So they are not good competitors, because when bacteria and fungi have done their work, then actinomycetes comes forward. And fungi, the more difficultly decomposable substances accumulate as they face less competition from the other organisms. Okay, they are not good competitors, they then decompose. They are responsible for decomposition of the more resistant or organic matter. Please note these points, they are very conceptual, they are really important for our knowledge. So please remember them, and produce several dark black to brown pigments, probably contributing to the dark color of the soil humus. Then next is some actinomycetes are human, animal, and pathogenic, like Microbacterium tuberculosis. The second is Mycobacterium, not Microbacterium, Mycobacterium tuberculosis. Second is Mycobacterium leprae. Second is Actinomedura, and fourth one is Actinomyces and Nocardia. These are some pathogenic actinomycetes. Next is they form symbiosis with non-leguminous. It is important, with non-leguminous, like some species fix nitrogen in genus of Frankia. It is a very most important term, right? Frankia. So actinomycetes are in symbiosis. In which example? So we can learn the Frankia. We know actinomycetes are not fungi because of one critical difference: they are prokaryote. They are different from fungi. Why? Because they are prokaryote, not the eukaryotic, right? So they are prokaryotic cells. Actinomycetes produces the metabolites that can make the smell of fresh plowed soil. You must be knowing this line. If you are a student of agriculture, this line is the most famous line I can say about this soil biology. In soil microbiology, this fact is most famous. Every student knows that the smell after the rainfall is due to the actinomycetes. But most of the students don't know that which type of substance cause this smell. So you should know that the geosmin substance, I have also mentioned that, but I don't know where is that point, but I will be mentioning that point on the screen, so you can write them. So please note that geosmin, Geosmin, geosmin substances are secreted by actinomycetes that produces a fresh smell after the rainfall. Okay, so please note that point. And musty, cellar, and Dracula's castle, these points are not important, but this point is important: smell of freshly plowed soil. Okay, so some important examples like microspore, these are already I have mentioned those names, and you can remember some important examples of actinomycetes. Next is viruses. Okay, so these are important topics related, important facts related to the actinomycetes that can be directly asked in the various examination. And the very most important point was geosmin, which was unfortunately not mentioned on the slides, but I will surely be adding them onto the screen, so you can write that also. So now focusing on the viruses, you must be knowing that viruses are the smallest microorganisms. They are smallest microorganisms. They are only 20 to 100 nanometer or smaller in size. Okay, they are smaller than bacteria, and then they are the smallest. You should know that. Consequently, it requires an electron microscope to visualize viruses. Viruses need higher quality of microscope so that they can be seen easily. As far as we know, viral bacteria are inert. What is means of inert? Inert means they are non-living outside any organism. They need any organisms to survive, right? They are inert and do not carry any biosynthetic function outside the body of any organism. So please note that they are inert and they do not carry any biosynthetic functions. A virus are obligate intracellular parasite. What is that? Obligate means they are completely dependent on as parasitic relations, right? They are intracellular parasite. They cannot grow or reproduce outside the host, right? They can survive outside their host and remain infectious. They are outside the body of any organism, but still they are infectious, because if they get entered into the microorganism, into the host cell, then they will cause infection. They are considered non-living. Why? So viruses are the link between the living and the non-living. Actinomycetes are the link between fungi and bacteria. Please note these points. The non-living, by some within a favorable environment, they behave as living cells, right? Outside the host body, they are non-living. Inside the host body, they are living. That's why we are saying they are the link between the non-living and living. And viruses are sometimes ecologically, but more often environmentally important because of their capacity for diseases. Okay, we will be also discussing some important, I will be showing some important ecological benefits related to the viruses also in this slide. So here are some famous pictures of viruses that you have already seen before, like HIV, hepatitis B, Ebola virus, and the bacteriophage. The bacteriophage is the most famous virus. I think everyone is familiar to this diagram. And herpes virus, rabies virus, and etc. Okay. Next is the more famous viruses, coronavirus and bacteriophage. Bacteriophage structures are important for the studies purpose. So let us discuss now the life cycle of viruses. There are two main life cycles. The first one is lytic and the second one is lysogenic. Lytic is also known as the virulent. Virulent, it means they are dangerous. They completely kill the host cell. Lytic means completely break down. Okay. So what is the meaning of lysogenic? Lysogenic means they have capacity to break down. They have capacity to kill the organism, or we can say the host cell, but they not surely kill that organisms. So but this host cell are for sure dead by the in the lytic cycle, but in the lysogenic cycle the hosts are not always killed. Okay, so we'll discuss about this more. Here is the Lytic cycle. There are 5 main steps that are asked in the various semester examinations also. So please note that the first one is the attachment. The second one is the attachment. Attachment means those attachment that we get with our friends. No, sir. Don't involve the emotions or feelings in this video. Okay. We will not talk about that. Attachment. Attachment means the attachment of bacteria to the attachment of bacteriophage to the bacteria. Bacteriophage is a virus that kill the bacteria, right? So the bacteriophage get attached to this bacterial cell wall, which is known as the attachment. The second is penetration. Penetration of what? Penetration of genetic material. Bacteriophage transfer its genetic material into the bacteria, which is known as the penetration. Third one is the biosynthesis. Biosynthesis means before the biosynthesis, the DNA of the host bacteria get completely destroyed. We can also see this into this diagram. Like in this step, this is genetic material of this bacteria. The complete genetic material is disturbed. It is destroyed because of the enzymes called the lysozymes and various enzymes. The bacteriophage kill or destroy the complete genetic material of the bacteriophage, and then the maturation phase. In this phase, the various type of parts related to the bacteriophage, various parts like the capsid and all, we have already seen these parts in the capsid, sheath, tail fibers, spikes. These parts get synthesized in this phase. Synthesis of viral genome and proteins. Various parts of virus are made and they are assembled, and the new viruses are formed. When the new viruses are formed and they get mature, they get completely synthesized, when they are converted into new viruses, then these viruses get released from the cells by collapsing the cell. So we can say there is a lysis. Lysis means the complete destroying of the cell. So the new viruses will be released into the environment. So this is the lytic cycle where there is a complete destruction of the host cell. Okay, next is also you can learn these steps from this diagram, and I have also mentioned them as theoretically. And there is a lysogenic cycle. See, lysogenic cycle can be attached to the lytic cycle also. Can be attached means in lysogenic cycle, I told you that there is a capacity of the bacteria, or I can say there is a capacity of virus to kill or lyse the bacteria cell. But you can say seen that the DNA is not destroyed as like the lytic cycle. In lytic cycle, you have seen the DNA was completely destroyed of the host cell. But in the lysogenic cycle, a part of bacteriophage DNA, a part of bacteriophage genetic material is attached to the genetic material. Attached to the means here was some, as you can in the blue, there is a genetic material of bacteriophage which is get attached to the genetic material of bacteria. Then if there is environmental conditions, if there are some favorable conditions, then it will get into the lytic cycle, or then it will be get destroyed as in the lytic cycle. But if it is just about the lysogenic cycle, then it makes more and more copies of the DNA, or I can say the genetic material in which the part of bacteriophage is present. If there is a part of bacteriophage DNA in the host host genetic material, then these cells are called prophase. Please remember this term, that it is important, which is known as the prophase, and they get multiplied again and again. And if the environmental conditions are good, then lysogenic cycle get continue as the Litic cycle, and the viruses uh, get outside from the cells. So this is more simpler diagram related to the Litic cycle and lysogenic cycle. And if you are preparing for the agricultural examinations, these are not so important. The virus part is not important. The actinomycetes part is very important. But if you are preparing for the some other examinations, then these are important topics. These are also very important for the biology students. These are virus cycle are very important. Okay, I tried to make you understand these cycles, but if I am unable to, if you don't get these topics, then please watch other videos on YouTube also for more clarity, or you can also read these topics, as theoretically I have mentioned these topics. So at last we'll be focusing on the ecological roles of virus. Viruses are important as they control the microbial population. As we said, bacteriophage can feed on bacteria, so they can control the population of microorganisms. They are biocontrol agents of gene exchange. In gene exchange, viruses play very important role. Sometimes excision, human pathogens, and virus therapies. These are some other points that are related to the viruses. So in next video we'll be focusing on the fungi, or I can say the soil fungi. Okay, so thanks for watching this video. I hope this video will be helpful for you, and I will again say that the topics or the points related to the actinomycetes are very important. So please don't avoid them. Please kindly learn them for various examination, or I can say any examination that you are preparing for. Okay, so see you in the next video, and I would request you to please like this video and share this video with your friends. And for more such videos, stay connected to the channel Agricultural Studies, and bye bye, take care.